This is a production of Cornell University.
Thank you very much. Pleasure to be here at Cornell. I think this is my first time. I'm going to use an old-fashioned pointer so that the folks in Geneva can see me. Can you see me in the pointer here, guys? Can I can't hear them, right? Anyway, I hope they can hear and see.
They'll wave their hands.
They'll wave, yeah. Are they waving their hands? So, so what I'm going to talk about is actually kind of a simple study that we thought was going to be simple when we started, and it turned out to uncover some very interesting aspects that we had not anticipated about something about xylem structure and function. Now you may think that xylem that's been studied for a very long time, it's a fairly well-known structure of different kinds of cells, right? There's vessel members, there's tracheids, fibers. I'm assuming I don't have to introduce these concepts to all of you plant science folks here in Cornell. And all of these are arranged in a tissue, and of course different species and different groups of plants have different kinds, okay? But one thing that was known from early on is that the structure of xylem is fairly complex. These things are not just pipes that run straight. They're pipes that run crooked. In this case, this is about a 2 millimeter by 2 millimeter by 30 centimeters, 35 millimeters section. So it's sort of been compressed vertically so that you can see that these things kind of wander around. These are vessels. You can see that they're interconnected. And in fact, this section and this section, this is all the way back from sort of the 70s, need to overlap one another to actually, they're from the same section. They're just separated so that you can see there's a whole lot of spaghetti in here. And we had some questions about xylem movement that we wanted to ask. And one of the things that a complex series of interconnected pipes will do, which is something that I did not know before I talked to an engineer, any engineers in the audience, is that you can actually, if you have 3 pipes, and one goes straight through, one has a bit of a resistance, and another one has a bit more of a resistance, and they're interconnected in this fashion, that even though water may enter into this side on all 3, going that way, and water may exit on all 3 on the other side due to a pressure differential, part of the system may be flowing backwards. Is everybody okay with that idea, that you can actually have backwards flow even though it's coming in and going out the same 3 tubes? So this would be called, I think, an emergent property of an interconnected system of pipes. And that's a very interesting property because we were wondering whether reverse flow in this kind of system might explain how vascular pathogens, that is xylem dwelling, in our case, do they know about Xylella fastidiosa here, the cause of Pierce's disease in grapevine? Anybody know about that? It was all made the big headlines in California a number of years ago. It's a xylem dwelling bacteria that is transmitted by a xylem feeding insect, and usually the insect feeds in shoots in the upper part of the canopy, but over years the infection can get down into the roots and kill the plant, basically. It's called Pierce's disease. And of course, if it's in the xylem, how is it going backwards when the xylem is always supposed to be flowing, towards the leaf? So that was somewhat of a mystery, how a disease can spread downward, but maybe if there's some of these that are flowing in reverse, that could account for that. Maybe these kind of interconnections, whether it's downward or crossward, if we understood the interconnections, then we could explain how embolism spread. I'm assuming everybody here understands a little bit that the xylem system is under tension. Water under tension can cavitate or embolize and sort of suck air, let's say, cause an air bubble to occur in there, and that can spread. and obviously wreak havoc with the system of transporting water. So we had a couple of these questions, and then we asked, well, do we know about how this system's interconnected in order to answer these questions? And we thought, well, maybe we don't know. So we were able to get a grant and harvest a very good postdoc, Craig Broderson, who is now got another postdoc at UC Santa Cruz. And we sent him to our friendly neighborhood advanced synchrotron night light source in Berkeley. This is what Berkeley looks like all the time. Just kidding. It's a nice picture of the San Francisco Bay. You know, where all the men are strong, all the women are beautiful, and all the scientists are above average. But it's a big place inside there. That went over much better when I was talking in Minneapolis the other day. But you can put a plant inside here with this X-ray source, and you can get a very high-resolution X-ray tomographic view, a three-dimensional view of the stem. Now, this is a live plant, and we didn't start with live plants, partly because this is an air stage and it has to rotate in a very precise manner in order to get a 3D image, and it's a lot easier to do that if all you have is a stick of wood. And we were only interested in the xylem structure, so we began by just putting sticks of wood here and rotating them around and getting this information. And I'll get back to the living plant in a little while. But so what we get with that, so I guess, Neil, I forgot to tell you, you should have brought some popcorn, but chips are probably just as well. So here's an example that I'm going to just let play of what the raw data looks like. If you scan through this section, as we're scanning down, this is across 5 millimeters. This is the pith, and you can see that the pith is sort of changing. This is a dry piece of wood. These are all the xylem vessels, okay? Now I'm going to stop this and rewind it. So lots of interesting things. The pith goes from being full of tissue to being empty, but that's not what's important. What's important is what's going on in all these xylem vessels, and what you see is the big ones, and they don't change much. But next time I play this now, just take a look at some of these places where there are small cells. Just pick a spot. I mean, anywhere you pick, and I'm going to play, and what you're going to find out is that these small cells kind of dance around. We had a lot of fun doing this when we were, and you see they appear and disappear and sometimes connect over and sometimes move over, and there's a couple over here that just sort of shift entirely. Okay? But anyway, so the idea there is that we get raw data that is just basically a stack of images. They used to make movies out of this, and it took a long time. This doesn't take so long. And you get a very high resolution. And you can start to see then how the sort of pie-shaped pieces between rays, because that's what you have here, these are aligned up you know, radially in files, and that sometimes there are vessels that go from one ray and connect over to another ray, and a lot of times there are small vessels that interconnect these big vessels, and there's a whole lot of complexity in the way these vessels are interconnected. And that's what we wanted to try to model mathematically. Okay? Now, so this is just an example of one cross-section with the pith labeled here. Now, the grapevine bears leaves on either side of the stem, of the shoot, and those are called the lateral sides. And because these are called lateral sides, something has to be dorsal and something has to be ventral, I guess, even though those are terms we don't normally use for plants. But that's what this is called, dorsiventral and then lateral. And the lateral sides, you may be able to see a little bit here, look like they're a little squished in compared to the dorsal ventral side. Maybe the vessels are a little bit smaller than here, and we'll see a little bit more detail about some of those properties that they have. But as I said, they're sort of in radial files separated by rays. And this just explains that, in fact, every pixel of this is about 4.5 microns. And so we have 4.5 micron pixels this way, and also 4.5 micron pixels this way vertically, so that they're called voxels since they're a volume. And in order to analyze this, you can't just do it on image J. You have to do it on some kind of software, and this vivo software is kind of like image J on steroids. You can do all kinds of interesting things with this software. So I'm just going to sort of take you through a little quickly the process that we would go through to analyze a section. So we'd start with one slice somewhere in the field, and then as I click down, you'll see things change here, and this is just keeping track of all the processes that you've done, and the image will change depending on what that process was. Now the first thing you do is you kind of blur out the in-between parts, and that you see something showed up here and you see almost no difference there. I don't know if you noticed that, but I'll just go up and down a couple of times. It just kind of gets a lot smoother in between the cells. You just kind of smooth things out. Now, I have actually no idea of all the image analysis stuff that's being done, but I can explain what the results of it are. Once you've done that step, you can try to sort of segment this into black versus white, and you can play around with settings to make it a little bit more white. And then the nice thing about this software is that when you pick a spot here, for instance, if you just click on this, you know, the magic wand idea, I click here, then it will sort of connect all of that same color, basically, which is black and white, so all of the white that's not bounded, and it'll connect it up and down from wherever you clicked it, and then it'll also show vertical sections that through this thing if you want to. So it's sort of a volume-oriented 3D reconstruction software. Anyway, so we have to have somebody go through and click on these things and say, yeah, okay, that's a vessel, that's the one that I want. And what happens is you can take two of these things, or one of them, and you can identify it and give it a name, I think, and then turn it into just that. You can tell the software, okay, only show me what I've named here. And once they've been named by this guy, Jacob, that's his name, he's the undergraduate who was doing all of the driving of this software, then you can generate a volume rendering. So this is just a quick movie to sort of show, and you'll see right here a lot of dancing around the vessels as it scans through. And then basically once you've identified them, you can get sort of 3D renderings that you can rotate around, and it's a lot of fun to watch. But because it's done mathematically, you can do virtual cross sections this way, and because of the volume filling, you can actually identify these and then have it tell you where is that volume. And in addition to the volume, it can tell you where the midpoint of it is, where the edges of it are, and how close it is to its nearest neighbor. So this was all preliminary, because what we wanted to do was we wanted to find out how these things are interconnected and make a model out of it. So for instance, but this software was very helpful in being able to show when, for instance, you had a connection because they were close together, and when there was space in between them, when they diverged. When you're looking through a cross-section, you may see it in this way when it's at one part of the stem, and then farther apart when it's at another part of the stem, and you can volume render that and see what it's trying to tell you. But now we had the question, well, how close is close enough? Okay? Or if they're this close, then are we going to consider them connected or not? Okay? And another question is, when you have a connection, then what's the hydraulic resistance for water flow through that connection? Well, that actually can come from the literature, because some people have already looked at that. But what we had to do is we had to go back to the drawing board a bit and make a measurement on a cross section. Here we have introduced into one vessel at the end of the stem what we called a fiduciary marker, and the marker is just some silicon resin, so that when we made a cross section of that and put that in the SEM, we could find out where that was. Then we could identify from that which vessels were close to it. Then we could look under the SEM at detail, and when you see a pit between two vessels, then you know that they're connected. Okay? So these are typically, you know, the typical pit fields between vessels. They have a pit membrane or middle lamella across them, and those have been pretty well studied. So this is just showing the vessel-vessel pits over here and the vessel to parenchyma pits over there. So then what we could do is we could look at all of the cells, how far they would be measured apart in the X-ray image, and whether they or not they had pits with adjacent vessels. And this is a graph showing anywhere down to about 8 or 10, 12, 14. By about 14 microns between, then the amount of cells with pits between them, the amount of vessels with pits between them dropped off sort of precipitously. So we figured if it's 14 microns or less between, we're going to say that's a connection. If it's greater than 14 microns, that's not a connection. So that's how we kind of calibrated the distance, the information we could get from the X-ray from the anatomical information about whether to call it a connection or not. Any questions about that so far? So one of the things that we discovered was that we saw some of those vessels that sort of dance between big vessels, the little ones that dance between big ones. Actually, we looked at those, and those are actually open. So I don't know whether they'd be called a pit or not. Anyway, but that was something we sort of discovered late, that even that it may not be the typical vessel-to-vessel pit that has a pit membrane and has a resistance across it, but rather an open pit. Those might turn out to be those will turn out to be interesting in a later. But so once you've decided how close they need to be connected, then you can let this software go through and not only trace the center of the vessel, but also trace where there were connections. And this is all kind of off-the-shelf automatic after you've gone through that process I described. But you can see sometimes it thinks that there's connections right in the middle of nowhere, basically, and a lot of these connections looked really crazy. So we ended up having to hire another postdoc, Eric Lee, who is an engineer type guy, and he was able to develop the algorithms that we needed to take what a vivo software told us and actually turn it into something that we thought was more realistic and that he could actually model as far as flow was concerned. And then we went ahead and checked checked what the data looked like in terms of where the connections were and so on. So one thing, for instance, was if you consider a vessel where 0 degrees is in the radial direction and 90 degrees is 90 degrees from that, the connections are mostly at 0 degrees with some at 90 degrees. And in the dorsal-ventral zone, same thing in the lateral zones. But this made a lot of sense because generally vessels are in radial files, so this would be a 0 degree connection. And they're also usually next to, if they're going to be connected, they'd be connected at right angles to that rather than at sort of just any old angle. But that was so that's just one description. We had a bunch of descriptions. You know, that the dorsal ventral vessels are generally bigger than the lateral vessels. They're closer to the pith and more interconnected. But all of this is just descriptive. What we really wanted was to take all of this interconnected stuff on a particular stem or a particular section of stem and make a mathematical model, a flow model, an engineering model about how water would flow through this system, because that's what we were really interested in. So this is what Eric did. We took a bunch of information from the literature about, for instance, what the pore size distribution would be in a typical pit, what the pit conductivity would be, and a bunch of other stuff, and came up with a flow basically from one pressure at one side to another pressure at the other side, just to see how this would behave. And this is the first ever run that we did. And what this shows is, in the red color, it's flow going in one direction. It actually doesn't matter which one, but let's say from the bottom to the top. And the blue direction and its intensity is flow going in the opposite direction. So already in this particular section that we analyzed, we've already got enough complex interconnections and enough differences in resistance and so on that you can get flow in the wrong direction in a reasonable model of what the xylem would be like in a grapevine. So that was one of the things we wanted to ask, the question about whether you can have reverse flow, and you can. Another thing has to do with embolism spread. I'll get to embolism repair a little bit later. But is there any pattern that you can see that relates to the structure of the xylem, or do things just embolize, let's say, randomly? Well, here's an example of a living stem now, and notice that you don't see many of the vessels, and that's because there are a whole bunch of other vessels that you can actually pick out by eye, but they are almost the same color as the background because they're filled with water, and X-rays are basically telling you the density of the tissue, and that's mostly the density of water. So basically all of these vessels that you see here are embolized vessels. And this was on a stem of a plant that we dried out on purpose, then took it to Berkeley, then set it up in this X-ray, and then X-rayed it for a number of hours, and got sequential images along this 5 millimeter section. And what the images showed was all the ones in blue here were the ones that were already cavitated, already embolized when we started, so at time 0. And through the rest of the 8.5 hours, you can see how it spread as the colors go. I can't see colors very well, but you can see a couple of embolizing here and so on. And one thing that you could see very clearly was that they tended to embolize in groups corresponding to that radial file, which makes sort of sense because those are the ones that are typically interconnected. But we had some cases where, so if you look down at this lower half, for instance, this group, which I think is red, I'm red-green colorblind, so is this a red color here? Okay. This group in red embolized at the same time that this group in blue, is that blue?
Yep.
This group in blue, and this is yellow or green?
Yellow.
Yellow, thank you. So we got a red file and a blue file and this yellow guy, and if you scan through, you find that that yellow guy switched allegiance here from the red to the blue somewhere in that section, and you can see in the 3D reconstruction it's going from being connected to a red guy to being connected to a blue guy here. So that explains why all of these things would embolize at the same time, is because they were connected by one of these, by the yellow guys. So I think in terms of sort of conclusions for this part of the work, we think that we can use high-res CAT scanning to construct accurate 3D models of how the xylem is interconnected. And it's a lot of work. It's a lot less work than doing everything by hand, but it's still a lot of work, and we're still working on that. I think the 3D software to rotate these images and make virtual sections through them is very valuable. We can see a lot of interesting things, like those bridging cells that we didn't even know about. And I think the idea of backflow and perhaps spread of xylem-dwelling bacteria may be an important, that the complexity of interconnections and the idea that you can have backflow may be important for that. And also that those 3D interconnections may be very important for the pattern of embolism spread. Any questions about that so far? I'm used to kind of interacting a little bit more with the class, so I really encourage you to ask questions. I know everybody's embarrassed. Sure? Yes, sir. Good.
One question. The gaps, the interconnections, what's the space there and how does that relate to the diameter of Xylella or other organisms that may be moving in that space?
That's a really good question. So let me see now. So if you were thinking about this gap, that gap is the I don't know, is the Bay Bridge for Xylella. Xylella can get through there no problem. That is essentially open. The pores in the pit membranes of a typical vessel-to-vessil connection, those are too small for Xylella. And the thinking is that the Xylella excrete enzymes that degrade carbohydrates, and they digest their way through those, probably causing a cavitation when that happens. I mean, but that's where the spread of the bacteria and the anatomy of the xylem and its 3D interconnections. That's kind of where these things converge to the same kind of question. That's why we wanted to model this. Actually, the model that I showed hasn't included these kind of connections yet because we sort of discovered those after we had figured out the model. Good question. Anybody else? Yes, sir.
How do you calculate the hydraulic resistance versus water flow?
That's a very good question. So, and I don't know. No. Well, so you hire a postdoc who knows how to do that. Then that solves it. So Eric is actually a postdoc in engineering, and so the values that you can get that from the literature about one value, for instance, is just a Poisson equation for a given diameter of vessel and the flow through that. That's easy, right? Okay. The hard one is how hard is it for water to go from here and go across when there's a pit connection? And as it turns out, there are ways. So you can take a piece of stem and you can glue, using super glue, of course, glue a glass capillary that's been pulled down pretty fine, glue a capillary into one vessel. Then you can send water with some dye through there and look at the other end and see where that's coming out. Now, usually what we try to do, then you can glue up that side, and if it comes out of an adjacent vessel, and the vessels are fairly close, then you can be pretty confident that they are connected in between there, and that then you can measure the flow coming in and going out and the pressure differential, and then you can calculate a sort of a hydraulic conductivity of that junction. So we've done a little bit of that in our lab, but other folks have done a lot of that in their labs. So that pit conductivity is something that we just put into this hydraulic equation, along with all the other typical assumptions of a hydraulic equation. Are you an engineer type guy? It's a very complex set of equations, right? Because you got all these vessels and all these interconnections. That's why we had to hire Eric to do it. Is that okay? Yeah. We had no idea how this would work out. We just said, let's just model this based on physical principles. Good question. Anybody else? How am I doing time-wise? Okay.
I have a question.
Yes, sir.
It's not obvious to me why there, from a pressure gradient point of view, why there is backflow at all.
Okay, so you should have stopped me way back when I was talking about this.
But you didn't say it was okay at that point.
Well, okay. Basically, if the pressure here at this connection, let's just say for a moment that this connection has very little resistance. If the pressure here is lower or is higher than the pressure here, then water's going to go back that way. And you can just make up a scenario. These are all equal-sized pipes with slightly different resistance. So if this one has some resistance, then the pressure downstream of this resistance is going to be lower than this pressure. And if this one has a lot more of resistance, then the pressure here is going to be a lot lower than that pressure. And they both may be lower than this pressure here with no resistance. So basically, this is really, from here to here is just a connection, and whether it's oriented backwards or whether it's oriented to the side, and to some extent doesn't really make any difference. So the water doesn't know that it's going backwards, it's just flowing downhill. Now that's just hand waving, but.
Well, I'm glad I asked the question, it works for me.
Yeah, okay, so apparently the engineer guys have known this, and if you are a young engineer and you design a system of pipes and this does it, you get fired or something like that. You know, I mean, that usually you don't want things to work that way, but I think that's why they always use pipes of the same diameter, because then you don't have that big of a problem. This is great. Anybody else? So let's see, we talked about embolism spread. So I'm going to quickly now talk just about this repair, because at first we were just putting sticks in the HRCT, and we decided, and I don't remember who was it, whether Craig wanted to do this or Mark Matthews, my colleague, said, you know, let's put a live plant in there and see if we can see embolisms or something. Now the guys at Berkeley said, you guys are nuts. You know, you don't go anywhere near that thing, you don't have anything living in that thing, you know, there's all the shielding and stuff, and they just thought the plant was just going to die. Well, it didn't die. In fact, the plants, we brought them back out to the greenhouse and they were fine. Just as background though, other people have used NMR or MRI, nuclear magnetic resonance or magnetic resonance imaging, to say that there's been vessels that are embolized. So here's a corn root, and you see these 3 black dots, and now there's only 1 black dot because the other 2 are full, and that means that, you know, that water has filled these vessels that were embolized. So this has been around for a little while, but as you can see, the resolution's not great. If you have a corn root with really big vessels, then you can see them. We actually got a little better resolution on grapevines, and we're also able to show, so this is a living plant, and you know, you can see them light up here, and where there was two dark ones here, okay? But again, the resolution's not great, and we put a living plant in there, and it survived. And for instance, we would make a CT scan of this plant that showed some dark vessels, that means cavitated vessels, and then you could look and see on the image that there were vessels that were water filled, no problem. And you could kind of line that up with a hand section of the same thing. So for instance, this is pointing to, number 1's pointing to 2 adjacent vessels here that we would say are connected, and the big one is embolized, the little one is full of water. Okay? But as soon as we started looking at these, we saw these little blobs, these little like warts, and we thought, what the heck are those things? I mean, most of the vessels, you know, are round, but we saw one that are like moon-shaped, and what are those blobs? Well, the thought was, maybe those blobs are little water droplets that are starting to refill these vessels. And Martin Canny had already come up with this idea. So as background, I probably should have said this already. You know that we get embolism. That's a bad thing. And there's been lots of evidence that embolism can be somehow reversed. So you can have a loss of hydraulic conductivity in the day, and then a gain of hydraulic conductivity either during the night or maybe sometimes later in the day. And so the search was on for finding out how the heck a xylem vessel can be empty at one time of the day and the whole system's under tension, and yet can somehow refill, you know, later that day or maybe overnight when the system's under tension. So Martin Canny had used cryo-SEM and found little drops and said, hey, maybe it's water being sort of pushed in there by the adjacent cells. But the problem with cryo, of course, is that you're never sure if it's an artifact or not, because you freeze it and water can distill and condense and so on. So that kind of got left alone for a while everybody was chasing after this. And also, these droplets, now you can see that they, in this virtual long section, that they look a little bit more like drops. They kind of look a little bit like tyloses, but obviously tyloses are a permanent thing. They are not a drop. It's an outgrowth of an adjacent parenchyma cell. And we basically just kept looking at these, and finally, once we got the 3D sort of reconstruction figured out, we you know, to our amazement, realized that what we were seeing was drops. And they may be drops that you could see clearly as individual drops, or they may be things that were sort of more big blobs, which may have been drops that just coalesced. So what we could do is we could look at the same section over time. So this would be an example of an experiment where we took a grapevine, dried it out real good, came in at 0 time, and this is just one long section from one section where you can see 3 pretty clear vessels. And then we would maybe re-water the plant. Or in one case, in the first plant we did, we actually just put it in there, and at time 0 we saw this, and then he could see that there was drops or something, so he just left the vine alone, didn't re-water it at all, and over time it refilled itself. So sometimes if the vine is the plant's very stressed, you have to water it and then it will refill. Sometimes it just does it, maybe it was ready to do it on its own. But you can see over time, and finally at about 4 hours, everything had been refilled. Now, you know, we don't have minute by minute. It's more like every 30 minutes or so that we can scan this. But clearly, things were getting refilled. And you can see a lot of interesting detail about, you know, one of the last things to go might be a bubble in the middle of the vessel or on the side of the vessel.
Is that 4 hours with sunlight or 4 hours in the dark? Or?
That's a very good question. This is 4 hours in the dark, but also because of the shaking because of the way you have to, you know, hold the plant. I think I showed because of the way you have to hold this plant, everything is sort of enclosed anyway. This one has got its head sticking up above
So the
this.
evapotranspiration.
So yeah, so you would expect
really low.
that it's low transpiration conditions for sure, for sure. So here's an example. For instance, if since the software will allow you to say, okay, I'm going to identify this vessel, or this vessel, or this vessel. Then you can, and then it will also say, I'm going to identify these things as drops, tell me everywhere that they're connected to, and then it'll sort of show you all those drops. Then you can actually calculate the volume of the vessel and the volume of the drops, and the volume by difference of the air. So this shows, for instance, some 4 vessels, and how they ultimately all had no air at 6 hours. And because we were able to track individual drops, at least in the early part of this process, we could say, how big is the volume of that drop now compared to how big it is 2 hours from now or 4 hours from now. And we could actually calculate a droplet growth rate. And we could take all the drops in all the surface area of that vessel and calculate how fast it should take a vessel to fill. And basically, the white circles are our calculation based on a drop growth rate. That's not crop growth rate, drop growth rate. On an independent sample then from what we actually observed for these things as to how quick the vessel could be, you know, filled up with water. And you can see really good agreement, little disagreement here, but basically the vessels seem to be filling in a consistent manner with what we calculate for a drop to grow. And in this case, there are some vessels that start filling, that is lower air volume, and then the air volume comes up and may wander around and may never go to 0. Okay? I'll show some details about that. So that's what we would call as refilling failure. Okay? So if we zoom in on this thing here, we see it here. Okay? So we see some drops here, some drops here, some big blobs, drop here.
Where is that water coming from? Adjacent parenchyma?
Well, I'll get to that. That's a very good question. So, from, I don't know, Fred's magic, it just appears. So in this case, for instance, we've got 0 up to 6 hours. At 6 hours, this one lonely vessel here, which is this guy, has not filled, but all the other ones next to it have filled. Here's a whole bunch of little ones and a lot of drops, and this whole group is here and it's full. So, and it's not for lack of trying, because by zooming in on that one vessel, you can see that there are drops growing. I mean, I'm assuming that now, by this point, that these things are growing over time and that they are, so the idea is, well, so this would be an example of where this filling attempt somehow fails and it ends up being completely embolized. But it makes sense if these things are filling and then after being full, they come under tension as the normal part of the xylem is under tension. Then maybe this one just cannot keep up with that and gets empty because of the tension in the rest of the xylem. Or at least that's the model. Just as sort of a sidelight, you can get lots of very cool pictures of this sort of 3D, and you could zoom through there and get seasick doing that. The droplets appear to be forming preferentially on the sides that are next to the radial parenchyma cells. And this is not a new idea. Other people have speculated that this was going on, but I think now we have sort of the first evidence that it does go on in vivo. And that is that you've got the radial parenchyma cells here, that it's probably carbohydrates, because if you girdle, you can slow this process down. That's not our work, but other folks. The only way we can explain it is that the cells somehow get, transport sugars or whatever into the adjacent xylem, that those sugars then osmotically from all the rest of this tissue draw water in, and that's what causes the water drop to grow. That you can get growth over time, that the growth, if it happens to connect to an adjacent vessel that's under tension, then that's probably not fast enough to keep that water from flowing out. But if it is fast enough, which means that this is where things get interesting, is that we don't think this system, when they're refilling, is all under tension. It may be that there's other xylem in other places that are under tension. And we certainly, when we measure the bulk of the tissue, we get a low water potential value, so we presume that it's under tension. But it may be that this is a very local phenomenon, and that when you've got a bunch of cells, they tend to do this in groups, all sort of trying to refill. That's very teleological, don't get me wrong, I'm not a teleologist, but that's, I think it's pretty much what we think is going on. That maybe even in the ones that we think are full, maybe they're also getting carbon from their adjacent parenchyma cells, and maybe that they're kind of relieving the tension by adding some pumping into it. And while they're being relieved of tension, all the rest of these guys can refill, and that after they're done filling, then the whole system can come back under tension. At least this is our hypothesis. We have evidence anyway that the drop growth rate can explain the refilling. We have evidence that drops congeal and the space gets less and less. We have evidence that bubbles can be right in the middle of the vessel, and then the next time we see them, they're gone. So presumably they've gotten compressed, and you need a little bit of, it can't be under tension for that to happen. We also have evidence that the bubbles can be near the edge and disappear, which could mean that the air is sort of going out into the apoplast somehow. But bottom line, I think the weight of the evidence is strongly favoring the idea of a sugar or some kind of carbohydrate transport followed by osmosis and just water movement. Yes.
figure out why those vessels might not be under tension. Is there any evidence to suggest that the end pores in the xylem, in an embolized section of xylem, could somehow be closed or become closed during that embolic event?
That's a very good question. Missy Holbrook at Harvard came up with the idea some time ago that the pits. So actually, it's not just the ends that are a concern. It's the ends and the sides and everywhere, right? If the whole tissue is under tension, then how is it that you can get water under maybe slight pressure, or let's say no tension anyway, filling these things, and as soon as it touches a side that's in continuity with anything under tension, it should just get sucked right away. So she came up with the idea that the reason why pits are designed the way they are is that there might be a little bit of a hydrophobic pocket right around the pit, so that when water is filling this, it actually doesn't wet the part of the pit that connects it to the adjacent cell, and that it might be just at the last moment when the pressure is enough to cause all of those things to make a connection. So in a way, you could say that that's like, you know, closing off the connections until they're ready to be connected.
This has been a production of Cornell University, on the web at cornell.edu.